Howdy, welcome to Horticulture Innovators, the podcast series where we will explore impactful innovations in horticulture. I'm Amit Dhingra, your host. I'm a professor and head of the Department of Horticultural Sciences at Texas A&M University. In each episode, we'll explore the inspiring stories of the pioneers who are contributing to sustainability, wellness, and food security through their amazing work in horticulture. Howdy. Welcome to Horticulture Innovators. Today we have with us Dr. Lorenzo Rossi, who's an assistant professor of horticultural crop physiology in the Department of Horticultural Sciences. Welcome, Lorenzo.
Thank you for having me, Dr. Dhingra.
Hey, I know you've been here for almost a year now.
Yes, a year and changes.
All right. A few more months.
A few more months.
And, plant physiology, crop physiology is one of the oldest sciences. We've been trying to understand how plants really work. And I think for people's understanding human physiology, it's almost like, trying to figure out how things connect, how they work, how they're interrelated as well. So what got you interested into crop physiology?
Well, that's a good question. So as a kid, I grew up in Italy. And as a kid, all my friends wanted to play soccer. That was the thing to do. It's Italy. So, but I didn't. I never really liked sports. I like going and watch sport, but I was not really an athletic person per se. And so I remember my mom was pushing me. So it was like, well, you need to do something. You need a hobby. So the next try was piano. She was trying to teach me piano. That was another big failure. Didn't work out. Even the teacher, like, ma'am, you're wasting time and money here. And then my mom would keep saying, so what do you want to do? And I told her, I want to be outside. I want to grow things. And as a kid, I was in elementary school. I think it was third or fourth grade. I will have my garden. I will grow tomatoes, lettuce, a little bit of cucumbers. And I remember I will plant the irrigation. I will dig like kind of ditches and everything to have the water going. And that's kind of how it started. And a lot of people made fun of me. All my friends they think I was a little bit off, but it worked out in the end.
Well, you were ahead of their time. I mean, they need to eat that food that you were already producing. That's amazing, actually. So was this field called horticulture there, or what was it called?
I was going with agriculture. That was the idea in Italy. We have hortofloriculture, but the department is hortoflorofruit. So it goes vegetables, fruits, and floral.
Oh, okay.
But as a kid, no, it was just
agriculture, yeah. And so what happened after that? How did you find yourself getting into higher education, or what happened during college time?
So in Italy, at the end of when you are done with middle school, you have to pick an high school. And there are different type of high school. I decided to go to a technical high school with this kind of track for agriculture.
Okay.
And that's where I learned more about olives, more about grapes, more about different type of crops. And after that, it was kind of natural to kind of continue in that path. So I enrolled with the University of Pisa, and the Bachelor of Science was Agricultural Sciences.
Okay.
And since I graduated with high marks for in high school, I was able to kind of get into an honors program with Italy. So I was doing my work at the University of Pisa, plus I was doing additional work at a small, it's called Scuola Sant'Anna, which will give me a full board. So I will get free food, free rooming, free everything, free lodging at college, but I will have to take additional classes. And actually because of that track, I was able to do a period abroad in North Carolina State.
Oh, wow. So that was during your undergrad?
I started during my undergrad the first time. I really, really enjoyed that, and then I came back during my PhD.
So was that like a study abroad program?
Yeah, it was a study abroad. It was 6 months.
6 months, okay. At A&M we do a lot of study abroad programs, and I know that some of our colleagues are taking study abroad groups to Italy and the Netherlands pretty soon, where horticulture is rather big. That's fascinating. So walk us through what happens next. So you did your undergrad, and then how did you get into your PhD? Where and what did you do?
I really like what I was doing. My undergrad, I had to do a capstone, and I decided to work on apples and peaches and antioxidant compounds, in how the environment influence the antioxidant compounds in apples and peaches.
Yeah.
And we found that, I don't know, the canopy, the more pruning, less pruning, the rootstocks, the cultivars, there are a lot of that into the amount of antioxidant. And I really like that. My professor suggested me to continue for a master, which I did. And my master was still on apples and peaches, but more on the carbon sink. So, and the idea that you can use canopy material that when you prune, to put it back in the soil and to kind of have your idea of closed loop. We didn't call it closed loop back then. It was more sourcing.
Sure.
And that was my master.
You know, two things you just said. You were working on plants and health already during, what year was that?
2006, 7.
So almost 20 years ago.
Almost 20 years ago. I started during my bachelor. I graduated in 2008, so with my bachelor,
Isn't it fascinating? Sometimes we do things in our earlier years, and now you're in a position, we'll talk about that, because one of our mission sort of goals for the department is wellness, where we talk about interacting with plants for mental well-being and obviously consuming healthy fruits and vegetables and nuts to keep ourselves healthy physically. So it's kind of interesting. It's come full circle, right? I mean, it's I think, a common theme amongst faculty over here in our department that there was some level of working with plants for either feeding people or, you know, for me it was how do you solve malnutrition as well. So that's fascinating that you just mentioned that, and I had no idea you were going to say that, actually. So that's fascinating. And then, so you finished your master's there.
Finished my master. I was debating if I should go for a PhD. I had an offer from a private company in Italy, really close to my hometown, and I was considering it because it was a good offer, and he was more like about quality of their it's a transformation. They will do jams and marmalades from citrus. So it was more like a quality or increased production. But I really like academia. I wanted to be an high school teacher as a kid. And so I was like, if I go to the private sector, if I go to the industry, I will never go back to academia. But if I go to academia, I can always go back to the industry, or I can have. So that what guided me through the PhD. I did a rotation the first year. The idea was I started with grapes, but I was not really. The idea was organic grapes and copper accumulation into the berries, but it was not really what I didn't click with the advisor, and so then I switched with olives and salinity, drought, and that's where I started doing all my root biology and plant physiology, and that kind of defined me as a plant physiologist.
You also mentioned previously about carbon sink. You know, I think there's a lot of buzz around carbon markets, carbon credits. I know that's not what you're talking about. Can you elaborate what carbon sink means for the general audience?
Well, the idea is that carbon is available into the soil, right? And plants are uptaking it, and kind of there is a sort of depletion of that, because carbon is not something that you kind of put it back with fertilizer. Fertilizer are mostly nitrogen, phosphorus, and potassium. So carbon is actually the major component of organic matter. And the idea of depleting the organic matter means that you are kind of altering the soil functioning, and you're just relying as heavily on synthetic fertilizers. So if we can kind of bring back, or if we can kind of replenish the carbon sink in the soil, which means that we will have more organic matter. And the organic matter is the base for all the nutrients uptakes, microbes interaction, root growth and development, and every water uptake. So that's the idea of the carbon sink.
And the original carbon comes from the process of photosynthesis, which plants capture, and that's how they build. So when you're talking canopy, it was not a nice canopy to give you shade. It's primarily for the plant. Leaf area index, which how many leaves are present per square area, so that you can get how much carbon you can capture from the environment. So that's what I was just talking about. So you finished your PhD then, and what did you study in that? Olives? Did you make them better or bigger or healthier?
The project has three different objectives. The first one was drought and salinity, and we were working on a 25 years old orchard in southern Italy. So I will travel there, and my idea was to understand the anatomy involved with different when you're under drought. Yeah. So we cut down the trees and we measure all the vessels in the different years, and we found that when there is a drought, the vessel, your vascular system, they will grow smaller. And if you think about this, it's if you're trying to drink from a cup with the big straw and you only have a little less of water there, you're not going to get all the water. Yeah. But the plants adapt and create smaller vessel, and so your physics, your capillarity, your tension, and so they try to uptake all the water they can.
Well, they also develop embolisms, right?
Yes.
They develop embolism as a bubble in the vessel, and that can actually kill that whole sort of cell. But that's fascinating how plants are so flexible in developing mechanisms to take plasticity. Yeah. I want to avoid the use word plasticity because we are going to talk about plastics later on. But just it's flexibility, but plasticity is the word. So that's fascinating that you were able to work with olives. I'm sure. How many types of olives grow in Italy?
A lot, and some of the varieties are old. They're coming from the Romans or even older. Oh, wow. So we're talking about 2,000, 3,000-year-old varieties.
Do we find any in the U.S.?
Some of them, but most of the Italian varieties don't do well here because they're not really made for intensive systems. They're all for traditional, like training system, which require a lot of work. So the more kind of cultivated varieties here in the country are mostly coming from Spain, Arbequina, Arbosana, some from Greece, Koroneki, and we have one mission that was developed in California.
Okay. And so there's different olives for oil production and different for stable. Oh, okay, just like grapes, so different for That's fascinating. So then where did your journey take you? And the reason I'm discussing this journey piece is because oftentimes a lot of students think that their path will be linear. We've kind of adhered to this concept somewhere that if you go through school, you go through college, you get your degrees, and then you will end up somewhere. The path is never linear in most of, so that's why I'm asking you
It
this.
was not linear at all. In college, at the beginning, I wanted to be an entomologist.
Oh, okay.
So I was in a track to become an entomology major. Didn't work out, so I went with fruit science. During my PhD, PhDs are kind of intensive programs, so I had my struggles and everything. With the idea that I will stay in Italy, I never thought about leaving Italy. I did for a little bit, but my idea was always to come back. Yeah. And during the PhD, my last year, I wanted to go molecular. So I did anatomy, as I told you. We were doing a lot of physiology, but there were no kind of molecular component, like gene expressions or everything, to go a little bit more mechanistics and understand. And my advisor suggested me, well, you need to go to a different institution if you want to go that route. And that's what I did. I applied for several colleges, and I ended up at North Carolina State again because I know the professor, and that was an easy path. And there I did gene expression of, I think it was 15 genes related to salinity and olives, and I learned a little bit about everything related to DNA extraction, RNA, and.
It seems like your first passion is physiology.
Yes.
And just to help us understand, what do you measure? What do you study, and how do you study physiology of a plant? Or what aspects can you really investigate?
I'm interested in everything that kind of put an interaction between plants and the environment. And lately we're adding soil. So soil, plant, environment interaction. So we're seeing this increase on extreme climate event, a lot of different type of weather during summer, winter, and how cultivated crops are responding, and how can science help in not only understanding what's going on, but also making sure we have the tools to breed for species. And that's why my work is heavily connected with other colleagues here in the department. I don't think nobody's an island. We work together on several topics.
Well, when you talk about plant and environment, I think it's very, I mean, in the grape world, that's the terroir, which is the, your immediate environment, but that includes diseases, that includes non-disease environmental factors as well. That's fascinating. And don't you think we've as, because there used to be this effort of like, oh, there's drought that happens once in a while, or extreme heat happens once in a while, or you had those hot regions and USDA had these different zones of production. But now all of that is getting jumbled up because we can have a winter event in the middle of high summer and vice versa. So, I mean, I think physiology becomes even more relevant today to really guide how do we breed crops, how do we develop new varieties of like olives or grapes or pecans, or fruits and vegetables to really survive those environments.
That's one topic. And if you think about Texas, I mean, the state is big. We have a lot of different climate and microclimate, different type of soils, different type of crops. So for a physiologist, this is like a candy shop. Everywhere I go, I find something that I can work on.
Well, according to USDA, 11 different growing regions are here, and then 1,300 soil types. So that's fascinating. It's a candy shop, but also it gives us a unique advantage in the world to grow anything we want.
Yes.
I mean, people think of Texas just as hot, but if you start going up in high plains,
but then you have drought too, and you have the desert horticulture. You have temperate horticulture, subtropical horticulture.
No, it's fascinating. This is the world's best lab to grow any kind of crops, actually. So after that, after you were done your PhD and you came to North Carolina, what was next for you?
I stayed there almost a year and a half. I finished up my dissertation there, and again the idea was to go back. I was never really thinking about staying, and I told my professor I'm ready to come back. I want to graduate, and my advisor in North Carolina and NC State suggested me, well, why don't you stay for a postdoc? I will keep you. I was like, I don't know, I want to go back. But at the same time, my advisor in Italy was telling me, no, you should stay. There is not much here at the moment, and it's a good opportunity. So I stayed there. I started a postdoc there. I stayed there for 1 year. And then that postdoc ended. It was 1 year contract. I wanted to do something different. And there was an opportunity here at Texas A&M to work on pollutants, which I found was something new and interesting.
What year was that?
2015.
Oh, wow. What pollutants?
We were working with cerium oxide nanoparticles and everything that was related to nanoparticles and nanofertilizers. And so I ended up working in the civil and environmental engineering department here.
Oh, wow. Okay.
And I realized that they didn't know anything about physiology. So, and they didn't even know how to grow plants, of course. But they were really good on the engineering part. And my idea was, well, I will not be able to be successful if I don't partner up with someone in horticulture. And that's where I found Dr. Leo Lombardini. And we started a long collaborations. And so I will be in this department some days of the week and in civil engineering some other days. And Dr. Lombardini was nice. He let me use some of the greenhouse. We did some research together. We did coffee, and he also has a Lycor. So I was able to use it.
So Lycor is a measurement device that captures, what does it capture?
It captures stomatal gas exchange. So the amount of vapor and water, mostly oxygen and water, that goes out and in and out from the leaves during the photosynthesis. Also capture the health of the leaves in terms of photosynthesis, how much kind of CO2, oxygen is going in and out from the leaves.
And a big shout out to Dr. Lombardini. Now he's now the department head at University of Georgia, Department of Horticultural Sciences there. He used to be here. We miss him here, but so you were doing that postdoc. So you were at Texas A&M at that time, and then what came next?
It was a postdoc. So as all the good things come to an end,
yeah, that's right.
I start applying for jobs, and because of this research I was doing on roots and drought and salinity, one of the things that kind of defined my research was I was not just doing anatomy, but I was trying to understand how root kind of change their anatomy and plasticity when they are in contact with salinity, with heavy metals, with pollutants. And we found that they develop barriers at the, well, there are several layers in the roots, and I don't want to go into all the details about anatomy, but they kind of put a layer of wax, a waxy coating.
Oh, okay, around the roots,
on inside it's called the endodermis.
Yeah.
So they develop this layer, and if you use a dye, a fluorescent dye that react with the suberin, which is the element, the wax, you can visualize them under the microscope, and you can see that if you have pollutant salinity, this is there earlier.
Sort of an indicator.
Yeah.
It's fascinating that you brought up the roots thing, because I think if you look at the studies, or at least maybe I was blind to it, but a lot of the work was focusing on above ground. And because we can't see the roots, so they were both invisible naturally, but also scientifically, because we would not consider the root system sometimes.
And it was hard to study. I mean, in order to study the roots, you will have to dig the plant, or you have to excavate, and it was destructive. And in the past 20 years, I think we have been developing novel tools to study roots. We have mini rhizotron now, these transparent tubes that goes into the soil.
So tell a little bit. So it's called a rhizotron.
Yes.
And what does it do? So you have a column, like a plastic column.
It's a plastic tube.
Yes.
Plexiglass.
Plexiglass tube. Okay.
It goes into the ground. We usually put 45 degrees, so you can capture more of the root system.
So do you just put one tube, or you put?
It depends on what you're trying to get.
Okay.
We always suggest 2 or 3 tubes
per plant.
Okay. So it's more representative.
Yeah.
And then there is a special camera.
Oh.
It's a root scanner, and you insert the camera, and it take a 360 degrees picture.
So how does the camera move around the tube? Is there like a space between?
Yeah, there is a little bit of space, so the camera is able to rotate inside the
cool.
tubes, and you can take picture at different
levels.
Depth. Yeah.
That's fascinating. But how do you distinguish the roots from the soil?
So there is a software that we use, so a little bit of artificial intelligence there.
Okay.
And that can recognize by the color the roots and the soil. And then if you take pictures every month, let's say, you can reconstruct root growth and development over time, considering that you're taking the same picture in the same spot.
But also, I think although you're still constricting root growth, because root can go anywhere in nature, right?
Yes.
There's nothing. But this is the best approximation, perhaps.
It's the best approximation, non-destructive.
Yeah, non-destructive.
In the field.
Yeah.
For what we have at the moment.
Well, you're studying a very hard system, which is hard to track, but very important. Obviously, everything happens at the root system as well, or not everything, but half of it happens at the root system, half happens above ground. So you were looking for jobs, studying this part, and where did you start your career?
There was a root biology position at the University of Florida, and I didn't consider myself a root biologist. I was always thought of myself as a plant physiologist, horticultural crop physiologist.
Yeah.
Fruit physiologist for a short time. But I applied, and I did the interview, And the position was with the University of Florida at the research center on the East Coast.
Okay.
It's a beautiful area. It's on the East Coast. There is a lot of nice restaurants, and the center was also really nice. They were dealing with a big problem, which is citrus greening.
Yeah, yeah.
And so the idea was to increase root and soil health. So there is no cure for the disease, but if we hire someone that can help us, like managing the health of the soil and the roots, maybe we can get 3 or 4 more years
from before replanting.
Yeah, before replanting.
Are people replanting there?
They are, some of them, but the production dropped 98% in the past.
98%.
98%.
By some numbers, that used to be a $12 billion industry. Maybe more.
Now I think it's 4. Yeah.
It's probably dropped quite a bit. I mean, so talk a little bit about citrus greening. How does that disease manifest itself, and what's the function of the roots in there?
So it's a bacterial disease.
Okay.
If anybody is familiar with Pierce disease.
Yeah. In grapes.
Yeah,
Xylella.
Xylella, or olives too.
Olives has that too.
Yeah. Sweet cherries, I think they have something similar.
Yeah, we can get in pears as well.
Yeah, pears. Yeah. So it's a bacterial disease that it needs a vector. So if you think about malaria, is the same with the mosquito that will transmit malaria to humans.
Yeah.
In that case, the vector is a small fly that is called psyllid. And when the psyllid and the bacteria kind of get in contact, the bacteria will stay inside the psyllid body, inside
its
the
gut. Its gut, yeah.
And so every time the psyllid goes and feed on the tree, it will transmit this bacteria inside the phloem. It's a phloem limited. It's not xylem like the xylella.
Oh, okay.
So every time and if you think about Florida, we're talking about a mostly subtropical state, which no real winter besides some part in the north. Yeah. So the psyllid is active 365 days over the year. Yeah. And it feeding constantly. It's not just one time. We're talking about every day constant population of psyllid feeding.
Yeah, so it's non-stop assault.
Yeah.
So you talked about phloem. So just for everybody's knowledge, phloem is where food is transported to the roots from the leaves or to the growing regions. And xylem is where you have the water and the sort of, that's how water reaches the various parts of the plant. So that's from the root
water
system.
And nutrients.
Yeah, other nutrients as well. So if there is any obstruction in xylem, for example, in case of Pierce disease in grapes, plants will die. And in this case,
the phloem. So you will see that it's called citrus greening because there is no transport of all the sugars and other products that from the leaves needs to reach other part of the tree, and so you'll see that the tree is going to look greenish, it's going to look yellow. You start producing a lot of starches to defend himself from the disease, but in 5 years he will eventually be dead.
So there is no cure for that yet?
There is no cure. It's not culturable in the lab yet because it's a phloem limited bacterium. Yeah. And if you cannot culture it, it's really, really hard to
study it.
That's why we call it Candidatus Liberibacter asiaticus. It's a candidate.
Oh, okay.
There is a Liberibacter americanus, Liberibacter africanus. We assume that is the asiaticus.
Oh, okay. Wow.
But we cannot do the postulate to confirm.
As part of land grant systems, we try to support our farmers, but sometimes nature can beat you. And I think this is an example. I mean, there have been other diseases in history, like the potato blight.
American chestnut blight.
Chestnut blight. I mean, sometimes there are no cures for chestnut blight. Now they have a GMO version to counter, but nothing has worked so far in citrus greening.
There is a little bit of CRISPR-Cas going on.
So gene editing.
Gene editing. They are working, still working on citrus, of course, toward more acceptability of GMO orange juice.
Well, Texas also produces a lot of citrus, although different types, right? Mostly grapefruit. Yeah. So I know that citrus is a nationally important fruit crop, so that's very important. Are people trying to grow olives in Florida?
They are. I was actually part of a state project on evaluating the feasibility of growing olives in Florida. And we found a couple of spots that may be suitable.
As we are trying to grow them in Texas as well. But the freeze gets them every time.
And that was the same with Florida. I mean, if you find the right spot in terms of temperature, because most of the state doesn't have enough cold chilling hours. But the northern part, it does reach the amount of cold that is needed, but also it gets extremely cold.
Yeah. What I find so cool about plants, and there's a little play of words here, it's so cool that they can measure how much cold they have accumulated or how much heat degrees they have. I don't think people walking around picking fruit realize that if there was not enough cold or heat degree days, or so, you have to be below certain temperature or in a range at low temperature or at a higher to really make this system work. And it's all tuned with solar cycles. I mean, and the days. It's just fascinating how plants have attuned themselves to their environments. And now with this extreme weather conditions coming in, it is really going back to that whole...
Sometimes just 1 degree will mess up an entire flowering schedule or cycle.
Or if it's too hot, the pollination doesn't work.
Mm-hmm.
Or too cold, the flowers freeze. So it's a very fragile system. The food system is very fragile in that sense.
And it's a system that has evolved over millions of years of domesticated plants.
No, it's fascinating. So this is what you were doing in Florida. What came next?
Well, I was looking for something that, I always looking for something that would give me more energy or kind of put me to the next step. I think I was getting too comfortable. I like to get out of my comfort zone.
Yeah.
And I saw this opportunity for an horticultural crop physiologist. It was around the holiday break in 2024, I think. That position opened. And I had a lot of conversation with myself about should I apply or not, because I was about to get tenure there.
Yeah.
I already submitted all my documents, so I was kind of, my research was going well. I had good funding, but I was missing the teaching component. I was missing the interaction with a more like kind of academia community, because I was at the research station. I was not on main campus. And so these positions opened up, and I did apply. I was like, well, I'm going to try. If it works, it works. Now I already have a job.
Yeah.
And it worked. So I moved here actually it was December 2023 when I applied.
December 23,
Not 24. During 24, we did the interview process, and then I moved here in 2025.
Well, that was a strategic move for the department to really bolster our mission around sustainability, wellness, food security. And part of it was we really wanted to make sure we had plant, like exceptional plant physiologists in the department, horticulture crop physiologists, or people who work on secondary metabolism also, or plant breeding. And we really wanted to really bring in early career scientists in that field, in these 3 areas, to kind of continue supporting our industry, training undergraduates or graduate students and educate, having the latest in classrooms. As we are now moving towards robotics and mechanization and AI, we're trying to bring in those colleagues as well in the near future as well. So now we were very excited that you were here. And so while you've joined here, what has become your emphasis? I know that despite the fact that we move from our hometowns to universities, or we move from universities, the arc of what we study or what systems we work with doesn't go away, because we learn from these systems. And I also believe these plants choose us rather than us choosing them sometimes.
So I brought some of my research topics with me. We have a project with the Texas Water Research Institute, and we're identified plastics and PFAS, which is called forever chemicals, in woody crops. We're working on pecans and grapes. I'm still working on my the idea of soil health. I have some collaboration about the use of biochar for different type of soils. We're about to start a collaboration with Texas A&M Kingsville to kind of also continue my citrus research. And I also expanded, like these are all fruit tree crops. I'm expanding a little bit into vegetables and herbs. So
well, that's fascinating. That's going to be a great support for our industry on one side. On the other side, I think this is new material for our undergraduate students to be exposed to in classrooms, because you teach as well. I'll come to that in a second. And then also the graduate students who will train, because part of what we do in academia is pass on the baton. And I think that's needed in farming as well, because our farming generation is aging and we need to have the next generation come in. So that's our responsibility to train experts in that area. So talking about research, so you're doing this PFAS work or forever chemicals. So what do they do to the sort of ecosystem, first of all?
So those are long chain kind of residues that will stay in the environment, and they're small. We're talking about nano. And they're coming from different sources, plastics or everyday kind of trash, and they will find their way to the groundwater, and then it's not just one time, is the accumulation over time. And there was a study that tested different type of groundwater in the state, and they found high level of different type, and this can be uptaken by the plants when you do irrigation, and they will find their way into fruits, but also interact with the physiology of the plant itself, because it's something that can alter photosynthesis or nutrients uptake.
So it's not only impacting the accumulation piece, or which we will consume, of course, it affects us. Do we know all the negative aspects of consuming this over a period of time yet?
It's something new, so sometimes you need an entire generation to understand the effect. So,
but can we just say, like, wave a wand and get done with these PFASs? Because how hard is it?
Well, there are strategies that we are studying. Biochar is one of them. Use of again, and I think it will go back to soil health eventually. Taking care of your soil.
And we haven't really found any microbes that can break these down yet.
I don't think so, or at least I'm not aware of.
That's fascinating that you're working on that. So while you're doing this work, what are you focusing on, the above part of the plant or the lower part?
We're everything at the moment. We are studying the plant as a whole. I learned that a long time ago, and you say the same, that roots are actually part of the plant. So I think as a crop physiology, you have to study the entire crop, not just the above ground.
So how do you study the root system in trees, like in a pecan? What would you do? I mean, you can't dig up the tree because these are like massive trees.
The idea of the mini rhizotron is one of them, so having these tubes. Root density, that is just sampling on a monthly or every season, the amount of roots that you have. We can collect a small portion of root. We have a root scanner in the lab, and we can check for root diameter,
so this
color.
Is like an image analysis software. What about I heard that just like we can use radar.
Ground penetrating
radars. What are those called?
Those are GPS, ground penetrating radar. The technology works, but it needs some fine tuning still. It will give you more qualitative data than quantitative. So it will tell you where most of the roots are, and it will give you a heat map with different spots. But it won't kind of tell you the exact.
I had read somewhere that because soil has a lot of organic matter, and roots are obviously organic, the carbon containing, it's very hard to distinguish between them sometimes, or there are a lot of false positives. But that's an area of really technological development,
That one, and I think the other area that we have here on campus, that's the plant phenotyping facility. I was lucky enough to have a student in my lab that actually works there. And we are working on grapes, different varieties and salinity. and again, we're doing above and below ground phenotyping, and they have a system is an MRI, magnetic resonance imaging.
Oh, but MRI was for medical
Yes.
reasons.
we that's a that's a low field.
Yeah.
The one we are using for plants, and they will give you a 3D reconstruction of the root system.
So that's non-destructive.
And that's non-destructive.
But I think you still have to grow the plants in a pot.
In a pot. So it's not really made for the field.
Yeah. Wow.
But still,
yeah,
it's something that I think we should be really proud of. There are a lot of phenotyping facility, but most of in the country, but most of them don't combine above and below in the same space.
Yeah. No, that's great. I think we've gone from destroying the plant to study things in plant physiology to going into now non-invasive methods where you can measure from the top, but also like in my lab we study photosynthetic performance, where we can really see how the systems are working inside an intact leaf as well. So technology's come a long way. Hopefully we can also start looking at roots properly that way as well. So you mentioned that you decided to leave the research center because you missed teaching. And I know that you took on a course when you joined, which had been taught by the previous professor for 47 years. So how does Dr. Lorenzo Rossi come in and fill in the shoes of a professor? Because that was one of the most popular classes for the longest time. In fact, even before I joined myself 5 years ago to the university, just me walking around in the general public, people said, "Oh, Dr. David Reed, do you know him?" I said, "Well, he's in my department, yes." So that's fascinating. So how did you approach this, and what have you done? What kind of innovations have you brought in?
So as everything I do, I always trying to think a little bit before, and I realized that I have two options.
Yeah.
I could have kept the class the way Dr. Reed had for 47 years.
Yeah.
Or I could have bring something new, something that was more about the way I wanted to do it. And I decided to go for the second route. So, of course, I couldn't change the syllabus, but I could change the way the information is kind of transmitted. And that was the idea. So the first struggle I had was that students now need to, they will come to class if they found the class interesting or if they found the class engaging. With the constant information that we get from YouTube, from a lot of other sources, it's not easy. And I probably would have done the same. I don't know if I'd been going to a class which is just traditional teaching. And so what I did was the idea of combining, of course, a part of more like traditional teaching, but also a component in which we do story time. So for each modules, I have a story time on a specific thing, like citrus greening is a story time. We did a story time on dates and how they get to the U.S., the entire story of Dr. Zwingli going to Morocco and bringing the first offsprings here. And then the other thing was I realized that a lot of students didn't know what horticulture was, or there is a lot of kind of confusion between horticulture and agronomy. And I realized that we needed to make clear what are horticultural crops, how horticultural crops are in everyday's life. And that's what I try to do at least every week. We do tasting. And
tasting of what?
Something that they usually don't try a lot, like we did artichokes.
Oh, okay.
Dates, figs. We did a different citrus juices, orange and grapefruit.
Which one was the best?
From Florida and Texas. They like the Texas the most, of course.
They're used to the Texas terroir, right?
Olives, different type of olives. And that will also expose them to the variety of cultivars, of fruits, of everything. We do a flower display, and they can play with, I don't know, we usually do lilies because they're easy, so they can see the anthers, they can see the stamen, they can see the ovary, and that kind of keep them engaged.
Yeah.
And I mean, they may not come to all the lectures. There are 30 lectures, but at least I would say they come at least to 20 on average.
That's great.
And I think the most important thing for a professor that is teaching a big class like that, we're talking about 356 in person and
Wow.
175 online.
Wow.
Is to have an audience. The idea of teaching to an empty room kind of remove the not remove, but kind of
Dampens the passion.
the passion, and then you kind of start questioning it yourself.
Yeah. So is that the second largest class on campus? Maybe third
It's
largest.
one of the I think it's the third.
Okay.
With Dr. Shiner.
I was going to say,
Hort
even the
416,
largest class is also from our department.
Yeah.
So horticulture is such a big part of people, but as you said, they don't realize. So what is horticulture in your mind?
Well, I always say it's the heart. So there is an artistic component, there is a scientific component, but there is also a business component.
Yeah.
It's the heart, science, and business.
And technology is coming in now.
Technology is coming in there now. It's been there, but now it's
becoming more predominant. Because I think one of our challenges is labor. And I think mechanization, robotics, in many other crops like apples and pears, cherries, a lot of things have started happening. But other crops, like our pecans, I mean, if you go to the west part of the state, they are using mechanical pruning. They have to, because you can't climb a tree that large. So not only west, everywhere else as well. So it's fascinating. So and so in this classroom, so you said 356 students in person, 175 online. Are these all horticulture majors?
No, that's the other beauty of that class that got me exposed to pretty much every major on campus. This is what we call a science core curriculum. So it goes with the Texas science curriculum that came down from the legislation.
Yeah.
And we have to assess particular thing, quantitative skills, critical thinking, teamwork. That's why we have projects. That's why I do tasting. That's why I want them to think critically about different issues.
I notice a lot of posts on Instagram of the class and how much students are learning. But I think what is most important you're teaching is where does people's food comes from. I think one of the challenges is that if you're not exposed to it, you think the food just appears on the shelf at a grocery store at H-E-B or Kroger's or whatever you have. And I think that what you're really educating people from other majors is where food's coming from. I think that's really critical for our coming generation to understand. And we need to protect that source as well.
I always ask them to write a reflective essay at the end, and this is the time of the semester in which they're turning in those essays. And it's fascinating to see how many of them start the essay, say, well, when I first signed up for this class, I thought this was going to be another biology class, or this is going to be another one of those classes that I had to memorize a lot of enzymes. But then, no, I learned that there are different type of, I don't know,
olives. Peaches, olives,
and we grow them here in Texas, and I didn't even know that Texas was this important nationwide for horticulture, because everybody think about cotton and soybean and peanuts.
And cattle. That's right. Well, horticulture combined is a $70 billion industry for the state, 70, 80. And, between fruits and vegetables, pecans, grapes, ornamentals is the largest industry here. People just forget that that's horticulture. And also, of course, now controlled environment production, which goes everything from greenhouses to indoor farming to space. I mean, horticulture is everywhere. That's fascinating. So in this classroom, you're really changing minds to understand what horticulture is. And our mission of sustainability obviously starts with profitability for our farmers. But I think the major part is how are we going to sustain this sort of economic ecosystem, and that's where our focus on students. So what you're doing is really critical in educating the next generation who are Texans and anybody from outside who's here at A&M to really understand that we need to preserve this system. We need to obviously support our farmers. And then finally, you're also working on the third leg stool of sustainability, the soil health and plant health.
So the environment.
Environment as well. So that's fascinating. So that's one class that you teach. What else do you teach?
That class has a lab component.
Oh, okay.
So we're talking about 100 students from
okay.
or 201 usually take the lab.
Yeah.
We have 5 different sections, and the lab it's usually run by the TAs. So those are graduate students that we train them with teaching and research, of course. And that was another thing that I inherited from Dr. Reed. He did an excellent job, of course. So we took what we had and I kind of tweak it a little bit. So we're making we did make some changes. So we have 13 labs.
Wow.
And in each lab, the students will set up an experiment, and it needs to be set up in a scientific way. So 4 or 5 replicates. Each student is there are tables, so each student is considered a replicate, and they have usually we have 6 tables, so 6 treatments.
Wow. Okay.
And then we do statistics, and so we do drought, salinity, water. That's one. We do different hormones. We now we're doing hydroponics.
Which hormones? Steroids?
No.
Just kidding.
Plant hormones.
Plant hormones. Okay, I just want to make sure. Although plants do make sterols, but yes, plant hormones, which are different, of course.
Yeah, so those are mostly plant growth
regulators. I thought you were making the plants buffer,
Plant growth regulators. Some of them they will make them a little bigger.
Yeah, that's right. Yeah.
And we now we're doing hydroponics.
Oh, cool.
We're doing LED lighting system with different light color, and they have to record the data. They will have to do statistic, and for each lab they will have to submit a lab report on their observations.
So, and these are students from different majors.
Again, yes.
Well, that's fascinating. So you're kind of giving them those basic skills, life skills, because if you have to make a decision, what is better, what is not. I mean, statistics obviously comes in healthy. It's very helpful as well. And I also heard you teach another honors course. Tell us about that.
That was an opportunity. As I told you, I always like to kind of explore new avenues. There was an email that came out when I first got here. It was February or March. And they were saying that we are launching a new program. It's an honors academy. I know that A&M has a long history with the honors academy, but they kind of redeveloped the academy around the 6 core values.
So, hold on. A lot of people outside A&M have no idea what the 6 core values, but they're part of everybody's life. What are those? Please tell us.
So we're talking about respect, excellence, leadership, loyalty, integrity, and selfless service.
Oh my God, those values are, they found the core of our being an Aggie.
Being an Aggie. And so I did apply, and I was selected for 1 year.
Yeah.
So this fall and spring.
Yeah.
And so the first one's Honor 101, and we focus on the first 3 core values: respect, excellence, and leadership. And for each of the core values, we read a book, and then after that, we will have a class discussion, and the students need to submit a reflective essay.
Okay.
So we did that on 101 and 102. Those are small classes.
Yeah.
Those are 1 hour per week, 1 credit, seminar classes, 15 students.
Okay.
But it was great because in a small class like that, you kind of develop a sort of relation, and the topic that were discussed, they were all about nowadays problems, and you can see how students are passionate about that and try to find solution.
Yeah, what I find really fascinating that a horticulture professor is teaching about relish, but I think that also reflects the culture that we should have. It makes us quintessentially Aggies, and I think that's kind of our identity in a way. So that's fascinating that you did that.
I did it. It was a great experience. I enjoyed working with them. The final presentation were last week, and they were all excellent.
Yeah. No, this is fascinating. So as we wrap up the whole thing, and being an innovator, you've been innovating all through, starting from growing your own vegetables when everybody else was doing everything else, to today, while you're foraying into teaching honors courses in the or honors academy courses, also trying to develop new methods. What do you think foresee some of the challenges for food production, our horticulture crop production, or just food crop production would be in the future?
I
or
think
today, actually.
water will be one of
yeah.
the major problems. We're seeing this in Texas every day.
Yeah.
We're running out of water for a lot of crops that we're growing. Pecans,
Yeah.
as you know. Grapes can be affected too. Cold and heat stress are becoming another major theme. Also because not just the regular heat stress, but the one that is unexpected, or the one that cold snap that it was not supposed to be there in June and then actually is there. And so these are the things that I think from a physiology point of view, but also informing the next generation of students, and also collecting data and traits that can inform a breeding program. Of course, we also have the bigger picture, growing healthier fruits and crops that can increase human health. And that can be related to physiology as well.
Absolutely.
So, and taking care of our soil, inputs. You know how much it is becoming fertilizer are the cost is under the roof. So, having a better notion of soil health, regenerative horticulture. I think those are
some of the
areas that
are
going to be our tomorrow's.
Well, it's great for our department, our students, and A&M that you're here.
Thank you.
We look forward to seeing more innovations from you, and good luck with everything, and keep doing your physiology and inspiring the coming generation. Thank you for being here today.
Thank you.
Thank you.
you.
We hope you enjoyed this episode of Horticultural Innovators. Subscribe if you want to learn more and spread the good word. Thanks and giga.